fourth digestive gland is liver. Remember, liver is the largest human gland. Its weight is around 1.2 to 1.5 kg and is exocrine gland. And is exocrine gland and is present just beneath the diaphragm. At one side stomach and the other side liver is there. And this liver is having four lobes. Those are left lobe, right lobe, quadrate and caudate. Left lobe, right lobe, quadrate and caudate. In these lobes, we do find many compartments, and those compartments are named as hepatic lobules. Hepatic lobules. There are many compartments in the liver. Those are named as hepatic lobules. In these hepatic lobules, the liver cells are there. Those liver cells are named as hepatic cells. Remember, pancreatic cells are named as the cells of pancreas are named as pancreatic cells. The cells of liver, hepatic cells. These are meant for the production of bile. But these hepatic cells are there in one column. They are there in the form of columns. Hence, hepatic cords. Hepatic cords. Hepatic cords. Hepatic cells are the liver cells. They are meant for the production of bile, but these cells are there in the form of a column named hepatic cord. Once the bile is produced by hepatic cells, is conducted through hepatic duct. Hepatic duct. If the bile is produced in excessive, it is stored in one sac-like structure named gallbladder. Gallbladder. And from these hepatic cells, bile will go to gallbladder and stored through cystic ducts. Cystic duct. Bile can pass through hepatic duct, but if it is in excessive, that is stored in gallbladder, going through the cystic duct, both hepatic duct and cystic duct together named as bile duct. Bile duct. And this bile duct carries bile before it opens into duodenum. It joins to pancreatic duct. Together formed pancreatic duct, hepatico-pancreatic duct, and it opens into the duodenum. Hepatic duct, cystic duct together form bile duct, and a bile duct from liver and a pancreatic duct from pancreas join to form hepatico-pancreatic duct that opens into duodenum and is provided with the sphincter named sphincter of Oddi. Sphincter of Oddi. Let us see the composition of bile. It is secreted by liver, and this bile, its pH is 7.8, means slightly alkaline. That gastric juice is highly acidic, 1.8. Saliva, slightly acidic, 6.8. And it is slightly alkaline, 7.8. Is above 7 and nearer to 7. And one more interesting thing about this bile is that it is the digestive juice which doesn't have any enzymes. It helps for digestion but doesn't have any enzymes. Which of the following digestive juice doesn't have any enzymes? The answer is bile. And what are the things present in bile? This bile contains cholesterol, phospholipids, creatine, bile pigments like bilirubin and biliverdin. Bilirubin is yellow in color and biliverdin is green in color. These are the two bile pigments. Remember, these bile pigments, they are formed from hemoglobin of dead and worn out RBCs. After 120 days, every RBC will die and the hemoglobin present in it is converted into the bile pigments like bilirubin and biliverdin. And next is bile salt. Bile salts. Bile salts are sodium and potassium taurocolate and glycocolate. Bile salts: sodium taurocolate, sodium glycocolate, potassium taurocolate, potassium glycocolate. These are bile salts, and it is the composition of bile. Remember, no enzyme in it. Bile pigments bilirubin and biliverdin, bile salts these, and cholesterol is also present in bile, and phospholipids are also there, and even creatine, aging factor, creatine is also there in bile. And this bile from the liver can come to duodenum. It is nothing but is a waste of the blood is separated in the liver and comes to duodenum. And only the function of liver concerned to the digestion is bile production. In addition to that, liver, it does various function like it is meant for the storage of food and also blood. Food is stored in the liver in the form of glycogen. Even is meant for the storage of blood. Even you must remember, blood is stored in liver. Blood is produced by red bone marrow cavity of a bone, and blood is pumped by heart, and at the end the RBCs present in the blood are destroyed in spleen. They are destroyed in the spleen. That's all. Concern 2. Blood is stored in liver. Blood is produced by red bone marrow cavity of bone. Blood is pumped by heart. And blood, the RBCs present in blood are vanished, destroyed in spleen. It is first function of liver as well. Second one, it is meant for removal of wastes from the blood, and it is having some specialized cells like Kupffer cells. Kupffer cells. These Kupffer cells are phagocytic. Phagocytic in the sense they can engulf the pathogen and kill. And these Kupffer cells, they are present in liver as well. Once in NEET, the question was asked, which of the following cells are present in liver? Liver cells are hepatic cells, no doubt. But even Kupffer cells are also present in liver. The cells present in liver are Kupffer cells. They are phagocytic. And the next, the interesting thing, that is, during protein deamination in each and every cell, there would be a formation of nitrogenous waste in the form of ammonia. But that ammonia through the blood comes to liver, and where that more toxic ammonia is converted into less toxic urea during a cyclic pathway taking place in the liver, that is ornithine cycle. Ornithine cycle. It takes place in liver itself. And the next one is, here I have mentioned in pancreas, glycogenesis. The formation of glycogen from glucose in presence of insulin takes place in the liver. Glycogenolysis, conversion of stored glycogen into glucose in presence of glucagon, is also taking place in liver. Lipogenesis. This is nothing but conversion of excessive glucose into fat. excessive glucose into fat, that is lipogenesis. If we consume more sweet, it is dangerous because that excessive glucose present in the blood is converted into fat. We become very fat and fatty, that is mainly because of sweet rather than fat, because this process is enhanced in the liver, lipogenesis, leading to the formation of fat, and will be stored. A stored fat that is formed from excessive glucose. That process is lipogenesis. During lipogenesis, the formation of glucose, fat from excessive glucose. And one more thing takes place in liver is hemopoiesis. Hemopoiesis, that is the formation of erythrocytes, means RBC. Even RBCs are produced in liver only in embryonic state. Only in embryonic state. These are all very important functions of liver. It is meant for storage of food and blood, removal of waste from the blood, and Kupffer cells, they may make the NEET question concern to this, phagocytic and ornithine cycle, glycogenesis process, glycogenolysis, lipogenesis, and important is hemopoiesis is taking place in liver, where the production of RBC, but only in embryonic state. That's all. Fifth digestive gland is intestinal gland. They are otherwise said to be crypts of liver column. That intestinal glands produce a juice named intestinal juice. Even its pH is 7.8, slightly alkaline. It is having many enzymes. Remember, all the enzymes present in intestinal juice are final step enzymes. When they take part in the reaction, that is the end of the digestion as well. In which the first one is enterokinase. This is the enzyme, it never acts upon any food. But what it does, it converts that inactive trypsinogen into active trypsin. Inactive trypsinogen into active trypsin. This trypsinogen is an inactive enzyme of pancreatic juice. It is converted into active trypsin by the action of enterokinase. Just like SCL converts pepsinogen into active pepsin. And this enterokinase is another enzyme. Just like lysozyme, both of them never act upon any food. That is antibacterial and is activating enzyme. Converts this inactive trypsinogen into active trypsin. And alpha amylase, maltase, sucrase and lactase, these are amylytic enzymes. And the erythrin having dipeptidase and tripeptidase, these two are proteolytic enzymes. Proteolytic enzymes. And intestinal lipase is lipolytic enzyme. And these two enzymes act concerned to nucleic acids. They are meant for the digestion of nucleic acids like DNA and RNA, along with nucleotidase and nucleosidase. One more, nuclease. They are concerned to the digestion of nucleic acids. This is the composition of intestinal juice.